Hey guys, how are you? Hope you're having a good day. I just saw period two a little bit ago. But anyway, we're going to talk about in this video viruses and bacteria. And you have the notes, the fill-in notes. It's going to be a little bit out of order because I'm going to talk about viruses first and then bacteria. And what we're really talking about is a lot of this is how they reproduce, bacteria definitely, and then viruses how they reproduce, but also how we categorize viruses, and how they take over our cell machinery to make copies of themselves. And the point of this is to know about them, but also because we use this information of bacteria and viruses when we do genetic engineering. So that's why really one of the main reasons I'm talking to you about this, so that we can kind of, when we look at biotechnology, we can kind of have a little bit more background. Alrighty. So viruses, they are obligate parasites, obligate intracellular parasites. And we say that they're non-living because they don't do some essential things that living things do. First of all, here's the how we categorize viruses and prokaryotes, which remember the bacteria. Viruses are not cells. They don't have the cell components. All they have is the genetic material, and they have their genetic material protected and enclosed in a capsid protein. And their main purpose is to invade our cells, take over, hijack our cells, and use our cell machinery to make more copies of themselves. So they do not use their own metabolism. They don't metabolize. They don't create their own proteins. They have the code for it, but they can't do it themselves. They don't respond to stimuli. They do multiply. They replicate inside of our cells, but they have to, again, use our resources in order to make new copies of themselves. And yes, they do evolve. We know that they mutate and they change. And we know that the coronavirus is doing that. That's the new strains we have. It is mutating also. And the new mutant form, the one that they're concerned about with the coronavirus, is it is able to, the part that's changing is the spike proteins. And what that is it's going to allow more entry into our cells. So it can get in better than the original variant of the coronavirus. So on your notes, I'm jumping right down to what is a virus. It's a microscopic pirate. It is associated with disease, and it is host specific. Now, it can host jump, like the coronavirus. It can jump from host to host, but usually it is going to recognize cell receptors in a certain organism. Or if it's a bacteria or plant, it's going to be able to inject its genetic information. But it is usually very host specific, but there are times that it can host jump into a different organism, like we saw with corona. Number two, they're obligate intracellular parasites. They have nucleic acids, which will mutate and evolve, but it requires a host. And they have no metabolism or other characteristics of life. All right. Okay, this is the size of a bacteria, and these are the viruses to give you a perspective of their size, like how big they are compared to a bacteria. Remember, the bacteria is about the same size as a ribosome, so that kind of puts it in perspective as well. So the origin of the virus, there's several different hypotheses. So it could have been around before cells evolved, or after, or during the evolution of cells, or it could have happened after. Did it evolve from a host, like when it was in a host? Okay, did it evolve from maybe the genetic material of a host? Did it somehow come out of that? Classification. It is characterized by size and shape, the type of nucleic acid it has, and if there is an outer envelope. So some have it, some don't. If they have an outer envelope, they have virally encoded protein spikes that allow them to gain entry to a cell. Okay, so that was number 4 that I just went over. Now, as far as what they have, the structure, they have an outer protein capsid that contains the genetic material, and I'll talk to you about what the different kinds are. It has a nucleic acid inner core, and it may have an envelope, which would have those spikes on it, or like proteins that would get it in, help it get into a cell. Okay, so it may have an envelope, and that sometimes actually gets the outer membrane from the host. So wherever the host came from, the original host that virus is in, if it is getting into an animal cell, it's going to come in through endocytosis, and it will keep some of that nuclear membrane around it. So that's where the envelope comes from. So if you think of like the coronavirus, it has that envelope with the spikes on it. That could have come originally from the host. Okay. So it could have all different kinds of genetic material. So it could be a single-stranded DNA virus. Let me get my pointer because that's fun. Single-stranded DNA virus. It could be double-stranded DNA over here. All right. It could be, and then from there, or we could have a single-stranded DNA virus. It says positive or negative. So if you are, if the virus starts off as single-stranded DNA, it will need to make a double-stranded DNA intermediate, and then from there make messenger RNA. It has to make messenger RNA. So if it starts as DNA, it has to somehow make messenger RNA, just like we do, because then it's going to it's going to have that code in its messenger RNA that we are going to, then it will use our stuff in our cells to make the proteins it needs. So that could be enzymes. It's going to need to make a new little capsid to enclose its nuclear material, its genetic material. So we also have things called retroviruses, and we use these in biotechnology. A retrovirus is a single-stranded positive RNA. And when I say positive, that means that the RNA is 5' to 3', and it doesn't have to be like turned into 5' to 3'. That is the coding sequence. So anytime you see positive here, it's the coding sequence that is ready to be eventually translated into protein. It doesn't have to be replicated into like backwards into the, it doesn't have to be replicated from 3' to 5' into 5' to 3'. It's ready to go. Okay. So the retrovirus is really special because, again, we can use this. It needs to take its single-stranded RNA, and it first needs to make DNA from it. So you can actually take an RNA and turn it into DNA, and he uses an enzyme called reverse transcriptase. Okay, so the reverse transcriptase, it works in reverse. It turns the RNA into DNA. Okay, so right here, this is not messenger RNA at this point, and it can't really, in this case, it can't really make messenger RNA from it. It doesn't act as messenger RNA like other viruses do. It needs to go to the DNA format first and then make messenger RNA from it. Okay, right here, the single-stranded RNA, it's a positive virus. It's already ready to become, to be translated into protein. Our coronavirus, not ours, the coronavirus, SARS, it is this type right here. So it is actually ready to be translated into the proteins it needs. Okay, single-stranded RNA virus, you would take that and need to make a positive strand to turn into messenger RNA, and then the double-stranded, you would use the positive strand of that. All right, so lots of different classifications. So I think I've given you guys the notes for all of that. So you may have to just write that on the side column because it doesn't look like there's really a place for it in the notes for all the reverse transcriptase and all that. So rewind and write that information about what the positive strand means, and how we need to use that retrovirus needs to use reverse transcriptase. to make DNA and then messenger RNA. All right, so the rest of the cells just talk about the coronavirus. It's got the envelopes with the spikes on it. And when we talk about the vaccine that they have produced so far that we're using is a messenger RNA vaccine. So they took the coronavirus, they got specifically the messenger RNA sequences here that just make the spikes. So they're not giving us the virus; they're giving us the messenger RNA. It's very clever how they do this, but they gave us they're giving us the messenger RNA sequence that is going to use our cells machinery to make spikes. So we're making little spikes in our body, and then the antibodies will recognize the real virus when it comes in. Okay, so imagine this is our coronavirus. It's going to act as messenger RNA. We were able to cleave out the parts that identify and cleave out the parts that will actually make those spikes, and that's what's in our that's what's in the virus. Okay, they put it in a little lipid protective enclosure, I guess you would call it, and then put the RNA inside of it, and then we will use it. It'll come into our cells and we will use it. It can go in, the lipid is going to come right into the cells, right? Because it is nonpolar. So it gets into our cells. That's what the lipid is for. And then we can start constructing those spikes. And there was something else I was going to say, but it is completely gone from memory. It was important, but maybe it'll come back. I don't know. All right. So I already talked to you guys about this. It could have evolved before the 3 domains, or it could have evolved from a host, because they are host specific. So maybe they did. Maybe they came from a host, since it does match the host. So remember, the 3 domains are archaea, the bacteria, and the eukaryotes. And this is just the classification, all right, showing you all those different types of RNAs. We classify them based on if they're single-stranded or double-stranded, if they have an envelope or not, and then if it's a DNA, that's the DNA viruses. These are all the little types of viruses up here, the ones we're familiar with. The RNA viruses is the second classification, different variations of the nucleic acid, if it has an envelope or not. Okay, so there's your positive RNA ready to translate, and it has an envelope around it with the spikes on it. And this is just more of how we classify. Okay, a couple pictures of the tobacco mosaic virus that is in plants, and then also in the tobacco plant. And then this is the influenza, the flu virus. And then to study viruses or to make vaccines, we can use eggs, because they will, if you inoculate them, if you put a virus inside of them, this is kind of what the virus does, like what you're looking at here in a way. It injects its DNA, or it's taken into a cell, DNA or RNA, whatever it is, or it's engulfed in a cell like us, because we have, we'll bring it in with endocytosis, and then it's going to start reproducing itself. So we can actually grow viruses this way for studying or using. All right, here's the coronavirus again. It's got the, here's a spike, the glycoproteins. Here's your little lipid bilayer. It's got the RNA, and then the little envelope protein also. Okay, so this is how this is the main characterization of the coronavirus. All right, so this is how viruses get in. Now, it's a little bit different in plants and bacteria versus us, because they have a cell wall. So animal cells, it's brought in, it's engulfed, endocytosis. The virus, the bacteria and the plants, it's a little bit different. It's going to inject the DNA into the cell. Okay, so if you look here, there's a little mnemonic, AP Bio Mr. A stands for attach. So in our cells, we have cell receptors, and that is how a lot of the viruses with some sort of protein will actually, they'll attach to that receptor, and then we will bring it in. And it's going to hide in there because it is like taking part of our cell membrane with it, so it can kind of sneak in. This is a plant or bacteria. This is a plant or bacteria. This is a bacteria because this is a bacteriophage, one that specifically infects a bacteria. First thing it does is attach. Then it penetrates. That means it's going to inject in the bacteria. It's going to inject the DNA or RNA into the cell. Yeah, this is a bacteria because here's a little plasmid. Biosynthesis, it is going to then take its, it's going to take that nucleic acid, whatever it has, whatever the virus has, and it's going to then synthesize all the parts of its new virus body. Okay, so it'll need to replicate its nucleic acid, whatever that is. It may need to make its, it may need to change from the positive, from negative to positive strand to make so it can translate that message or transcribe depending on what nucleic acid it is. And then it's going to start making the proteins for the capsule. It's going to make enzymes if it needs it. So if it is a retrovirus, it's going to need to make reverse transcriptase. So it's going to steal all of the stuff from the host in order to make that. It's coming in with just nucleic acid. That's it. It's going to mature. That means it's going to actually assemble the virus new bodies, and then it's going to release, which means it is going to leave the cell. And that could either happen by exocytosis or budding in animal cells, or it can also just lyse or kill the cell. Okay. This is illustrated in a, let me give you the notes for that specifically. So it says reproduction of the virus, attachment, it is host specific. It has a host specific match because it has to recognize that cell to go in. All right, it has to recognize something on that cell to go in. Penetration, it is engulfed in an animal cell or injected into plant or bacteria because they have the cell wall. Biosynthesis, it hijacks the host machinery. So a little bit more if you want to write this over to the right. With biosynthesis, it needs to make any enzyme or proteins for the capsid. So it needs to make its enzymes, it needs to make its proteins, it needs to make its viral components. All right. Maturation, it is assembling the new viruses. So it's making multiple copies, not just one. It's making a bunch of babies, a bunch of virus babies, because it wants to reproduce itself. But then when they leave the cell, they're going to invade other cells, and that's how we get sick. And then release, exit through lysis or budding. Okay, lysis is when it bursts. Budding is when it is going to fuse with the cell membrane if it is an animal cell, and exit. So looking at this, we have two different life cycles of the virus. The lytic cycle is the one I pretty much just described, where attach, penetrate, put the DNA in. This is a bacteriophage. This is in a bacteria. It's going to make more stuff so that in maturation, so biosynthesis, you make stuff, all the components. Then you're going to assemble in maturation. Then you release. Okay, so this would be, in this case, you're gonna lyse the cell. Okay. The other thing that can happen, and this can happen in bacteria or us, is if you can have the virus come in. So after it penetrates, it can actually be incorporated into the host's DNA. It can lay dormant for a while, like a cold sore will come out if under certain, like if there's UV radiation or something, or if you get a cold, if you get sick, it can be expressed then. So it was in there lying dormant, and that it can come out then. Another example of this would be like I think this is an example, I'm not 100% sure, but I think it is. Shingles is my assumption that this is what's at work, because if you had the chickenpox, usually in mild case of the chickenpox, that can actually, the virus can lay dormant in your body until you're older, and then you can get shingles. So that's shingles is just a virus that's been laying dormant for a while, okay, and it can be expressed later. So that's kind of incorporated into your DNA. I think that's how that one works. I'm pretty sure. So then if something happens, so like again, environmentally, if something happens, sometimes people could have these viruses dormant in their bodies. They get sick from something else, or their immune system kind of shuts down, puts them more at risk, more vulnerable. Then the viral DNA that's in viral DNA or RNA that's in our own genome would have to be DNA to be incorporated. So it's going to be making the DNA from RNA or whatever. It is going to be like environmentally something happens, and then it's going to enter the lytic cycle. Okay. While it's sitting inside of our DNA, that's called a prophage. Okay. Because it's not active yet. All right. So on the notes, the lytic cycle, it's a standard 5 steps. Host cell is lysed at release by a virally encoded enzyme called lysozyme. So when it leaves, it's going to use, if it's going to burst the cell and get out, it needs an enzyme to let that happen. So it's going to use the bacterial, I'm talking about bacteria specifically here because this is a bacteriophage. The bacteria is going to help it build this lysozyme, and then it's going to use it to burst from the cell. So the virus had the messenger RNA to encode for the lysozyme, and the bacteria gives it the building blocks, the amino acids. Lysogenic cycle. This is after penetration. There's an integration step where viral DNA is incorporated into the bacterial DNA. The virus is latent and is called a prophage, which is replicated as we replicate our own DNA or the bacteria. Environmental factors can trigger the prophage to re-enter the lytic cycle. HIV can work this way as well, and we'll look at that. An animal virus right here. Here's your virus coming in with the little proteins. It's going to attach to a receptor on the cell. Our cell is going to bring it in because it recognizes some of the things on the outside because this was already in some other cell, and it gathered that cell membrane. So we don't really recognize this as foreign yet. We don't have antibodies to it yet. Okay. Once we recognize this as foreign, we will make antibodies, and this will not happen, or it'll be very limited. Okay. So we need to be able to recognize it. That's why corona has been such a problem, because it was not seen by anybody yet. We had no antibodies. It was like a clean slate. So once that's recognized, then once we get the vaccine and we've made those spike proteins, then we'll recognize this and this will not happen. Okay. So you come in here, our cell brings it in, and then right here, we are going to then, this is where the virus tricks us into using translation to translate those, once it gets its messenger RNA, and the coronavirus, it already is, we're going to start making the products to make a new virus, okay? So you have to make an envelope protein, you have to copy the nucleic acid, and you're going to make a capsid protein, all from our stuff. And then it's going to fuse with the cell membrane, and it's going to leave, okay? So it's leaving with these little, what do you call it? Part of our cell membrane, and the next cell that it goes in recognizes it as not foreign, okay? So the animal virus gain entry in various ways. I'm on the notes. Some use their glycoprotein spikes on their stolen membranous envelope. This can be then uncoated, and the viral genome can enter the lytic or lysogenetic cycle. It can bud from the host using various membranes, a cell, Golgi, nuclear, or it could just lyse the host. It can lyse if there's enough virus copies made, it can just lyse, and then it just bursts everywhere, and then it can get into new cells. All right, so the viral spikes attach to our cell receptors to gain entry. This is additional notes. Viruses that enter animal cells take part of our membrane to hide them. They have genetic info, and they use all of our stuff to make their proteins. their nucleic acid will get translated into proteins. And again, those are the enzymes, capsid, and envelopes that they need. The retrovirus. Is that next? Yes. Okay. So HIV is an example of a retrovirus, and it needs to do a little bit that extra step because it is coming in as single-stranded RNA. This particular virus needs to first make double-stranded DNA in order to function. Okay, because this is like, even though this is single-stranded, positive-stranded, does not have the correct genes in it to give the correct messenger RNA to make what it needs. So it's going to use that reverse transcriptase to make DNA. It's going to use integrase to integrate into our into the viral DNA. And then eventually it is going to get into the lytic cycle where it makes the mature viruses. So a retrovirus starts as, well, I'm just going to give you the main notes and then a little bit extra. The retrovirus, for example, HIV, has an RNA genome that is converted into DNA first within the host cell by an enzyme called reverse transcriptase. It can remain latent for years. Antiretroviral drugs can interfere with a step of its life cycle. So antiviral drugs can interfere with the life cycle step in here. It starts as single-stranded RNA, positive strand, use reverse transcriptase to make double-stranded DNA. It has integrase to integrate DNA into the host, and it can be dormant. All right. So let's look a little bit at bacteria because this all goes together. So bacteria reproduce. Remember, bacteria have a single circular chromosome as opposed to us, and they reproduce using binary fission. So they're mostly just making clones of themselves. However, we can get a little bit of recombination genetically to give them a little bit of variation. And there's 3 ways that this happens. So I'll go back to the beginning of your notes now. So binary fission, that's where you make an actual clone of the offspring, and we're going to use that as an advantage in genetic engineering because we can, when bacteria grow, we do it on a little agar plate. It's like a little plate, and you can get little colonies of bacteria. Each colony is genetically identical to the other, which is going to be useful for us when we look at genetic engineering. So because the bacteria, I'm on the notes, because I'm on back on the very beginning of the notes, because it is haploid and has a short generation time, mutations are generated and passed on faster, and natural selection is faster than in eukaryotes. There are 3 ways that you can get some genetic variety, and I think we talked about this early in the year. One of them is conjugation, which is right here, where 2 bacteria come together and they can exchange plasmids. The plasmid is a little circular. Remember, these guys have 1 big chromosome, and they've got plasmids. Plasmid is an extra little piece of DNA that can be given to other bacteria. And it's an advantage to them because, like right here, this particular plasmid could be carrying a gene for resistance to some antibiotic like ampicillin. This is the reason why we we're starting to get tolerance or the bacteria becoming more tolerant to antibiotics because as we use them, we use them a lot, and the bacteria are kind of changing and mutating, and then they're passing on that mutation for resistance to another bacteria. So that's why they try to limit the antibiotics. So that's conjugation. They're going to form a little sex pilus under conjugation on your notes. Conjugation, sex pilus, Pillus, to pass a plasmid from one bacteria to the other. Transformation, we looked at that already. When we were looking at remember we were looking at the S strain, Griffith's experience with the experiment with the S strain bacteria that were lethal and the R strain, and how we mixed them together. He killed the S strand and the R strand picked it up and made it actually dangerous. So that would be transformation. And that on your notes, pick up new pieces of DNA from the environment. The host absorbs that genetic information. So again, we took the heat-killed S strain of bacteria, put it together with the R strain, which was not dangerous at first, and that just picked up the S strain and picked up that virulence, the dangerous bacteria. And then transduction is the last one. So here's a bacteriophage. When it injects its DNA into bacteria, it may pick up a little bit of the bacterial genome with it, like, you know, so once it gets in here, especially if it integrates and it gets into the bacterial genome, and then that virus leaves and infects another bacteria, it could take some of that previous bacterial genome with it and give some variety. So the transduction is on the notes, virus acts as a vector, which brings something in, brings in genetic information. And this happens from an infection by a bacteriophage. You inject new DNA from the bacteria it was last infected. It last infected. Okay. So getting into now, this is on your notes a little bit, not much, but I'm going to be talking a lot extra on this. So you do want to write down the basic information that I'm saying, okay? Because there's more to it than I'm just than is on your notes. Okay, so we're going to be looking at two main things here with this. Two really important things in biotechnology. One is recombinant DNA, where we can take DNA from two different sources and we're going to combine them together in a plasmid. And I'll talk about that specifically. And then PCR, polymerase chain reaction, is where we're going to amplify a gene. And then eventually this gets us into gel electrophoresis, trying to see if we have a particular segment of DNA. Okay, so this gets us into the crime scene stuff and where you look for a segment of DNA, paternity cases, transgenics, where you can do like genetically modified crops and all that. So we're using the fact that we can use viruses and use bacteria in order to do this for us. Okay, so I'm going to give you guys lots of different, we're going to look at this in class, like this little glowfish thing. But let me give you the notes first, and then, oh, you do have notes, never mind. So recombinant DNA technology, I'm on the notes now after the viruses. DNA is from two or more different sources. It requires a vector to introduce the rDNA plasmids with extra rings of DNA, and the viruses are commonly used. Two enzymes that we need to do this is we need restriction enzymes and we need a ligase. So I don't think it has the two of them there, but restriction enzymes and a ligase, okay? So for what's happening here is we are, when we talked about those plasmids, we are actually going to create a plasmid that we want. So the plasmid is going to be from a bacteria, and it can be taken up by another bacteria and start producing a gene in question. So for example, if we want to mass produce insulin, this is how it's done. We can find the insulin gene from humans. We can get a plasmid. We can genetically engineer a plasmid, and the plasmid is going to have in its DNA, it's going to have a little area where it recognizes the gene, our gene to make a plasmid, our sequence of DNA that will then produce that'll then produce the insulin product. Because we can get this bacteria. Then once we get the plasmid, we can get it into another bacteria, get it to take up that plasmid through transformation, and then it's going to start mass producing that product for us, okay? So this is kind of what it looks like. So here's your plasmid in the bacteria, and we can actually get that plasmid to open and insert a gene into it. Okay, so, and then we'll look at, and I'm going to have a little virtual lab for you guys to do, so you'll see how this works. But there are, here we go. Okay. So in any organism, we can actually cut the DNA, our DNA too. Sorry, I'm in the way, and that's not gonna, I don't know where to go. All right, I'm just gonna stay here. Okay. So bacteria, because they get invaded by viruses, by bacteriophages, they have a natural defense mechanism where they make these things called restriction enzymes that can actually chop up the virus. Okay, that's their little defense. They make an enzyme that will recognize certain sequences in the virus DNA or RNA, and it'll attack those at those specific sequences. So it's very specific. Okay, we can use that to our advantage. So because these are very specific, these enzymes, they will recognize a certain sequence of nucleotides. Okay, so let me see where we are at the notes. So I'm on recombinant DNA technology. Oh, it's there. Never mind. We're under restriction enzymes. Number one, these cut the DNA at specific sequence called the recognition sequence. The enzyme, the restriction enzyme, binds there, and it will actually cut the phosphodiester bonds on the backbone of the DNA. So what it's able to do then is this DNA restriction enzyme comes from a certain bacteria. It recognizes a certain sequence. So if you're trying to get a gene into a plasmid, the restriction enzyme let's say, for example, it's from E. coli. It will recognize a certain sequence on the DNA. It'll cut it on the plasmid, cut the plasmid open, and it will recognize that same sequence on either side of our gene that makes insulin. So it's cut specifically there. Then we can, since everybody's cut open, we can put that gene in with the plasmid, and then it'll seal using ligase, and that's going to incorporate that little specific gene into the plasmid. Okay, so there's two different cuts that can happen. You can have I'm on the notes again. You can create a blunt cut where it's all even, or you can cut with, you can get a cut that overhangs, and these are called sticky ends. So like right here, you've got this was cut. Okay, so imagine this is a little end from the plasma that was cut open by this enzyme. This over here is the end of maybe the insulin gene. All right, so these are sticky ends because they're like little broken parts of a ladder on each side. Then once you get that, again, this all happens in a test tube. Once you get those broken apart, then there's another enzyme that's going to seal them together, the ligase, just like the ligase that was used in replication of DNA. When we put everything back together after it was replicated, we use DNA ligase. Okay, so I'm on the notes again, to cut different DNA. So our DNA and the plasmid DNA with the same restriction enzyme. That means there's going to be complementary sticky ends that can be welded together. They're going to match. DNA ligase will seal the phosphate sugar backbone of the two sticky ends. Okay. So let me see. Let me get, okay, I think that's where we are in the notes. I'm going to talk about the next part. And again, I'll show you how this works because we're going to do a little simulation. This is where the cut is made, or where the bond is. So after you cut open the plasmid, remember it's a circle, you're cutting it open so you can insert a gene. And it's all based on recognition by specific enzymes. Okay, so it's recognizing both pieces of DNA, the same sequence in both pieces of DNA. Now, there's lots of restriction enzymes, so many. And they all come from different species of bacteria. So what scientists did was they took all of these enzymes once they discovered them, and they figured out which enzyme recognized which sequence. All right, so to do that, to find the gene that codes for insulin, they had to figure out which restriction enzymes from which bacteria were able to make cuts on either side of that that gene. All right, so they want to get as close to the gene as possible, and they're cutting on either side of the gene, and then they can stick that into a plasmid that they cut open with the same enzyme. They're going to match up the sequences perfectly. Okay. So here we go. Your restriction enzyme are biological scissors. They're also called endonucleases. They're going to cut at a specific recognition sequence. So here we go. It cut at that recognition sequence, and it's going to create these ends. So this was just one of your DNAs. This was that gene. This was your DNA. You're cutting out. You're going to cut out on either side of the gene right here. Well, actually, right here, it's cutting. It's recognizing this sequence. So this enzyme is recognizing GAATTC, and it's recognizing it backwards this way. So it's going to make that nice sticky end cut. Then once you get that, say, for example, the gene's up here, this is one area where it cut. Your gene is over here, maybe, say. There's another end up here that needs to be cut, and then your gene is ready to be put into a plasmid. Okay, they're named for their specific genus and species of bacteria. They're all specific. So, for example, EcoR1 was from Escherichia coli, the E. coli strain, and a specific strain, and they're all different. So we can use that. This is us using this knowledge to try to cut specific genes out of DNA. All right, and this is called recombinant DNA, where you cut out your, you put your gene into a new source. So for example, a plasmid. So this is what it looks like. This is a first genetic engineering experiment. So you cut out the DNA fragment of interest. I'm going to keep saying insulin because it's used so often. So for example, this was something cut out by restriction enzymes. And this yellow gene right here is, let's say, the insulin gene. This is a plasmid from a bacteria, and these can be artificially completely reconstructed now, and a bacteria will take them up. Why will they take them up? Why is this possible? Because of the universal genetic code. We all code for the same. We all have the 4 bases that code for all the same amino acids. So the vector is the recombinant vector is going to be this little thing with the gene that we want. Okay. So we're going to take that gene that's now inside of this plasmid. This is now a vector. It's going to transform. So you put it with a different bacteria or with a bacteria. And then what you hope is that the bacteria then is going to absorb this plasmid. And then it'll start dividing and look at all of the plasmid that you have produced. And then it will start making the protein that you want. So multiple copies of insulin. So this is where I'm going to be adding a lot. So you may want to type this, okay? So this is a big side note. In order for this to work, in order for us to take a gene of like from a eukaryote, put it into a bacteria and tell the bacteria to actually make a product that we want, like insulin, there has to be some things that happen. So let me make sure I wrote the notes here. So what you're trying to do from all of this, the goal is to make clones with the same sequence of DNA. You need to make the bacteria make that product. So if we are trying to make a whole bunch of insulin, the bacteria, and this isn't related to this slide right here. This is a little extra side note. The bacteria has to be given like whatever amino acids are going to that it needs to build insulin for example. So you're going to have after you complete the transformation, when you get the plasmid into the bacteria, you're going to feed it some nutrient broth that has the stuff in it to make the insulin, okay? So here, things have to happen in order for this to work. The first thing that has to happen is you have to have, on your notes, or on the last notes, you must have regulatory regions in the plasmid. So your plasmid, it's got to have a little spot that is those regulatory genes that we talked about with regulation of gene expression that are going to make transcription factors. It needs to have a promoter. It needs to have all that stuff. Well, the promoter is with the insulin gene, but it needs to have a regulatory region that can make transcription factors to turn on, to attach to the promoter and get that going. All right, so the RNA polymerase combined, you can make the messenger RNA that matches the DNA code for insulin, and then you can, so you need that regulation part of the bacterial DNA to make those transcription factors, and then eventually you'll get the, you'll be able to get the protein from it. So the RNA polymerase, it's like a switch, like we talked about before with eukaryotic or even prokaryotic gene expression regulation. The switch needs to be turned on. So just like development, the switch needs to be turned on to make a certain type of cell, to make sure that that is expressed properly. So you don't get that eyeball on your arm. So in this case, though, you do need the switch still. So RNA polymerase needs to be able to bind to the promoter site at the beginning of the gene for insulin. And so a regulatory region needs to be part of that plasmid. It needs to be transcribed and translated. So you need to have something made to bind to the promoter so RNA polymerase can start. Okay. Let's see. And again, this is all back from once you, this is after, this has to be in addition to finding the right bacteria or the right plasmid that can open up with a restriction enzyme that also cuts on either side of the gene that you're looking for. And then ligase, once you get that done, your ligase is going to, it's like a chemical tape to seal everything together. All right, so you've got that. You've got the regulatory site that once you get this thing into the plasmid, it'll make transcription factors that can bind to the promoter. You also have to have in your gene of question, so for example, the insulin gene, there can't be any introns in there. Everything has to be coding that you put into that plasmid. So the strategy for this, we're going to go to the virus now for some help. The strategy is we are going to use a retrovirus to make double-stranded DNA. So we are, you get your mature messenger RNA first, for that's going to code specifically for insulin. So it has to be mature. The introns have to be out of it and everything. Okay. Then we're going to use a retrovirus to make single-stranded DNA or double-stranded DNA from that mature messenger RNA. And then it's going to make a nice clean copy of the gene that's going to go into the plasmid. All right. So, and you need a vector. That's the plasmid that is going to get that gene in there. Once we do all of this, we want to make sure that the plasmid was picked up properly. So the simulation you guys are going to do has to do with a gene that produces a glowing bacteria. So there's a gene that codes for, it was from jellyfish originally, that codes for a glowing protein. And we can really easily see if the bacteria picked up the plasmid properly, because if it did, it glows. Okay. So to select for this, what they usually do is they need to make sure that the what they're seeing is actually the plasmid that has the proper gene in it, because sometimes the enzymes might not work well, or maybe the bacteria just didn't take it up. So you want to select for this carefully, and to do that, a really good experiment is just to look at this glowing gene, glowing protein. So what we usually do with this is we take ampicillin resistance gene is going to be in that artificially manufactured plasmid. And so you put that in the plasmid, And the bacteria is only going to grow if there is, if the plasmid was picked up properly because it contains the ampicillin resistance gene. So what they do is they can use, and you'll see this on the simulation, they can use a little plate, and I will have you watching an Edpuzzle too. They use a little agar plate, and they'll put ampicillin in it. So only the bacteria that took up the plasmid that has the ampicillin resistance in it are going to grow, and those will be the ones that are glowing. So that's how we kind of selectively see if we got the plasmid. So I wanted you guys to be familiar with mainly the restriction enzymes, because you guys are going to be doing your biotechnology thing. And those are going to come into play where we look at gel electrophoresis because they are specifically cutting DNA at certain sites. So if you've got like a bunch of criminal or people that you think are criminals, or suspects, you can identify which is which because you know what DNA was at the crime scene, and that's what we use gel electrophoresis for. Is we can cut that crime scene DNA with the same restriction enzyme because it's recognizing a certain sequence. And we'll do that with all the suspects to see who matches. So that's another example of how we use this. Okay, so that's it for now. And again, we'll talk about this as you guys are presenting your biotechnology presentations. All righty, so I will see you later.